In the last video, we looked at the fibroblast growth factor receptor 2 as a tertiary structure. That is, we looked at what the receptor would look like inside the cell, and it showed this blob type structure. In this video, we'll look at the structure more closely, and we'll be able to see the lots of little structures that make up this huge protein molecule, and we'll be able to see exactly where the 278F variant alters. If you look at this, this looks a little different to the blob in the other video, but you can see that it sort of follows the same structure. And again, we're only going to be looking at your immunoglobulin-like domains. So again, your immunoglobulin-like domain 1 is over here, IG2 is over here, and IG3 is over here. The variant that we're looking at is your cysteine 278F. And I've sort of got that highlighted over here, but let's look at the structures first. So these sheet type things that look yellow are called your beta strands, and these coiled red looking structures are called your alpha helices. And these are all connected by these green wire type things that we call your random coils. And it's when all of these join together that we get this huge protein molecule blob that we saw in the last video. When we have the C278F variant, it affects this region over here, and we'll take a closer look. It affects two building blocks that make up this huge protein, and that building block is at your 278 codon, and it's also at 342 codon. So at 278, you have this molecule called cysteine, and at the position of 342, you also have another molecule called cysteine. The molecule names aren't as important as the function as their functions. Okay, so this is what the normal structure looks like. When we have this variant, the cysteine at position 278, so at this wire over here, changes to a phenylalanine. And this is where your problem occurs. Okay, if we switch to over here, we can see it a little better. All right, if I zoom in on the two building blocks that are highlighted in pink here, this is what they look like. So the shape isn't important, but you will notice this yellow color bridge. And this is really important because this connects the two building blocks together. And when this connects the two building blocks together, it basically forms a loop that's present in all your fibroblast growth factor 2 proteins. And this loop is part of your immunoglobulin-like groove. So this loop over here is what makes this groove so stable. And due to this, these cysteine molecules are some of the most important building blocks of your immunoglobulin-like loops. When we have an affected individual and we have this variant occur, this cysteine changes to a phenylalanine. You can see that when this changes to a phenylalanine, these two molecules look slightly different. More importantly, when you have the two cysteines, they have a sulfur atom attached to them. And these sulfurs basically form this bond that we see over here that stabilizes this loop. When we have a phenylalanine, there is no sulfur here, only this weird looking structure. So it can no longer form this sulfur to sulfur bond. And when this happens, you don't have this yellow line joining the two building blocks together. So this loop is no longer stable. Because this sulfur bridge, or what we call this disulfide bridge, is gone, it basically leaves the cysteine at position 342 all alone. And this means that the Ig domain can now bind to any molecule that comes along, not just the fibroblast growth factor that it was binding to before. And because of this, the fibroblast growth factor receptor 2 protein can remain active all the time.